So this tutorial, we keep it as a complete tutorial that's going to be covering a lot of the details in the lecture part, and then Carson will be working through this tutorials for the worked examples. I find it very difficult to lecture off of a wiki page, and so I maintain a PowerPoint slideshow as well. It's essentially the same content, a little bit different here and there. If you want to follow along on the slides that I'll be doing, there's a link there to the slides that I just cut this morning as a slide share presentation, so you can follow along with that. But probably what you'll want to do is just watch the screen, and I'll share on the screen. Okay, so let's see, 2:22. I'm going to try to keep this lecture fairly quick, so let's get started, and we may leave a little bit out of the end of the lecture so that the tutorial can get started and Carson can fill in some details. Okay, so the annotation problem. First of all, probably everybody who's signing up for this course knows what annotations are, but just to orient ourselves, you can think of annotations in two different parts. We have the structural annotations, where we have the actual features of a sequence feature on the genome, so the exons, the introns, the start codons, the stop codons. And then we have functional annotations. So what do these things do? We've annotated that there's a gene at this location on the genome. The functional annotations add additional information about things that we can say about this gene and things that we know about it. The growth in genome sequencing, of course, has been exploding in the last 10 years. This is from 2012 because the Genomes Online database stopped making this data downloadable, but the problem hasn't gotten any better. This red line down here at the bottom represents the number of completed genome annotations in the database, and this blue line represents genome projects that have gotten started but are stalled or just in process somewhere in the sequencing or assembly or annotation stage. So a lot more projects starting than are getting completed. We're probably all familiar with this slide, this plot put out by the NHGRI. It shows the cost of sequencing relative to Moore's law. As many of you probably know, Gordon Moore was one of the founders of Intel, and he observed many years ago that the cost of computation was dropping at a logarithmic rate, at an exponential rate. So this is a log scale on the y-axis here, and this is the cost of computes over time. We were all pleased back in the early 2000s, in the early part of the decade, in the early part of the century, that the cost of sequencing was dropping at a rate equivalent with Moore's law. But then about the time the next generation technologies became commercially available, we saw a phenomenal exponential drop off of an already exponential curve in the cost of sequencing. And this is driving all kinds of innovation in our field, as we all know. If we overlay these two graphs conceptually, we can see that that gap started to open up at about the same time that the cost of sequencing started to really drop. And interestingly enough, that's about the same time that the first release of Maker was made available publicly. And so the goal of Maker is to help close this gap here. So over the last number of years, there have been plenty of annotation projects that have used Maker, including this beautiful cover story in Nature last year of the coelacanth genome, and most recently, the largest genome ever assembled and annotated, the loblolly pine, which was just published earlier this year. So just to recap a number of those projects, these are just some of the ones that have come through our group in one way or the other that we've had some kind of a collaboration or an association with, and there's a growing number of annotation projects that we don't even hear about until we see the papers come out in press. Okay, so we talked a little bit about the next generation sequencing and its impact on genome annotation and assembly. So the next gen genome project, genome annotation used to mean these massive international symposium, collaborations, consortiums that would have large budgets and large groups. The next generation genome project is typically even an individual lab or a smaller group. They're using short-read genome sequencing. RNA-seq data is often the source of evidence for where the gene models lie. Genome and transcriptome assembly are a whole new problem with this next generation data. After we get the genome and the transcriptome assembled, we go into genome annotation. Most groups will then go on to set up a genome database / blast server or what have you. And then they'll go through a round of manual curation with their community, perhaps get some more sequence data available, do a new assembly, and then they have to re-annotate and merge the old annotations with the new. So this whole second half of the next-gen genome is the realm that Maker helps with. We'll be talking about how it can help with that in this next section. First of all, a little bit about how Maker works. So if we think about where these structural annotations for a genome project come from, we have basically two sources of evidence. When we get a new assembly and it's just a blank slate, we have no annotations on it. On the one side, we can have ab initio computational evidence, tools like SNAP, Augustus, GenMark, FGenes-H, and the like. On the other side, we may have some RNA-seq, some protein evidence out of the databases, and most people who have been doing annotation for a while have come to find that the most accurate set of gene annotations, certainly as a starting point, come from some kind of an overlap. The ab initio tools do a great job, but they tend to over-predict quite a bit. Our RNA evidence is great evidence for where the genes lie, but it's dependent on having libraries that express the genes of all of the genes of interest, and we can't have an endless selection of RNA-seq libraries from all life stages and all tissues. And so we have to kind of combine these two forms of evidence, and that's what Maker does for you. Okay, so in the several slides coming up, we're going to be looking at these screenshots that come from the desktop Apollo web editor. And in this top section of this tool, we have, that's not showing up, so I'll turn it off. We have a black box that is going to be where we see the evidence and the ab initio predictions and the details that go into generating an annotation. In this light blue box down below, we'll see the annotations that Maker will be producing. On the x-axis across the bottom, we just have some hypothetical region of a chromosome in our genome assembly. We don't need these red and green dots up here at the top, but basically what they represent is the start and stop codons in all three reading frames. We're only viewing one strand right here. Okay, so the first thing that Maker will do is identify and mask repetitive sequences. Repetitive sequences, sequence repeats are a fascinating field, but they become much less fascinating when you're trying to annotate protein coding genes in your genome, because many of them will bear resemblance to protein coding genes, because many of them came originally from protein coding genes. And so, and even those simple repeats that aren't sourced originally from a retro element or something like that, they just tend to wreak havoc with the aligners and the tools that are going to help us predict where the gene models are. And so we want to get rid of those for now, maybe come back and look at them later. And so we'll mask those repetitive elements out. Maker will do that step for you. and run repeat masker and a couple of other tools to hide those repeat elements from the tools that will help you with the genome annotation. Okay, the next step that we will run is we'll run a series of ab initio gene predictors. You actually don't have to have an ab initio gene predictor run for Maker to generate some starting annotations, but running one, and in most cases you'll want to run more ab initio gene predictors, will give you a better starting set of annotations than not using anything at all. I won't go into the details of how these ab initio gene predictors work, but the vast majority of them are HMM-based, hidden Markov model-based, where they go through, look at the genome in light of a set of known annotations. So you always have to have some starting set of known annotations for your genome or a closely related genome. And they'll go through and look at all of those, and they'll treat the different parts of your annotations as states. For example, an exon or an intron. And as they read along the sequence of the genome, they'll observe in the known annotations how frequently you switch from one state to the other. They'll also note how frequently you have the individual nucleotides. Sometimes they'll analyze dinucleotides within the introns and within the exons and within the states as you transfer in between. And they'll learn from those known annotations based on that, and then they'll go ahead and apply that to the rest of your genome for looking for genes that look similar to what they observed in the known set in the part of the genome where we're looking for novel annotations. Oops. Okay, so then after we run those ab initio gene predictors, we start to align our evidence. This is the RNA and protein sequence evidence that's going to provide clues as to where the expressed, transcribed and expressed regions of this genome are. So in the case of RNA evidence, this can be any kind of RNA evidence. It can be ESTs. It can be complete cDNAs, and that would be wonderful if all of our genome projects had completely sequenced cDNA libraries, but we don't have that much anymore. And so these days, we're typically relying on transcriptomes assembled from RNA-seq data, and that's fine. It has its own challenges, but it can provide excellent evidence for a starting point for an annotation. And then in addition to the RNA evidence, we'll also often use protein evidence as well. When we get a new genome, we don't have explicit protein evidence for that genome typically, and so often what we'll do here is grab the proteins from a large protein database from SwissProt or NCBI or something like that, and all of those proteins and RNA sequences are aligned to the genome using BLAST or one of the versions of BLAST that's appropriate for the type of sequence we have. So BLAST does a great job at aligning a lot of sequences very quickly, but it doesn't do a great job. It doesn't explicitly know about the intron-exon structure of the kinds of data that we see as evidence in eukaryotic genomes. So both the protein and the assembled RNA-seq data will have those exons or those introns spliced out. And when we align those back to the genome, we recover the intron-exon structure of our gene, which is nice. But because of the way BLAST works, it'll just keep blindly aligning on past the intron-exon junction for a few nucleotides. BLAST hates to open up gaps, and so it'll try really hard not to open up that gap when it gets to the boundary, and then finally it'll be forced to open it up, and it may not close the gap back up at exactly the right place. So Exonerate handles that for us. Exonerate knows about the intron-exon structure, and it knows about splice sites, and so it cleans up those intron-exon boundaries. So Maker runs that step for you as well. Then most of the ab initio gene predictors out there are able to accept hints. So for example, if one of the tools, say SNAP, produced this gene model here that we're looking at up at the top, we might be able to infer from our evidence alignment either that this exon does exist or it does not exist, and Maker will go through and look at the initial gene model predictions that the ab initio predictors came up with. Then it will cluster that data together with the aligned evidence, and it will pass hints back to the gene predictors, suggesting that they include or exclude or extend or contract the boundaries of the exons that they identified, and ask them to come up with a new prediction. So this is better than Maker just randomly chopping those introns or chopping those exons out itself, because the ab initio gene predictor will actually pass that new evidence back through its hidden Markov model and kind of assimilate it in to come up with a annotation that it can still live with based on its HMM, but that better fits the evidence based on the hints provided by Maker. Okay, so after that, Maker does that and provides hints back to the ab initio gene predictor. The gene predictors will come up with a new annotation that is a synthesis of the hint that Maker gave it and its own HMM predictions. And Maker will analyze annotations from multiple different gene predictors and try to identify the one that is most consistent with the entire set of evidence that has been aligned for that region. And we'll talk a little bit more about how Maker does that in a little bit. But once Maker has done that, then it finally produces one consistent gene model, and by default, for a given gene region, it will produce just one transcript annotation for that region, unless the defaults have changed recently. That was certainly the behavior the last time I used it. And that's the intron exon structure for that mRNA. It'll add the UTRs, even though the gene predictors typically don't give you that kind of evidence. Maker can look up at or give you that kind of detail. Maker can look back at the aligned evidence and add the UTRs for you if there's evidence for it. In this case, on the 5' side, we didn't have the evidence for it, so nothing gets out. In addition to that, Maker outputs a number of metrics that can be useful in quality control and selection of your best set of gene annotation models, gene models to start with. Okay, so we can't see these down here too well, so to just highlight some of those metrics that Maker gives you: length of the UTR, the fraction of splice sites confirmed by ESTs, fraction of exons that overlap ESTs, length of other UTRs, number of exons, length of the protein, and so on and so forth. There's also an AED value here, which I'll talk a little bit more about in a few slides. So finally, when Maker has gone through that whole process, it's going to dump those annotations out in valid GFF3 format. We've looked in a couple of cases earlier this morning at what those GFF files look like, so I won't go into more detail now. And it also dumps out the FASTA sequence of the protein coding portions of those annotations. Well, not just the protein coding portions, but for the protein coding genes, you'll get files with your mRNAs, your transcripts in them, files with the proteins in them as well. Okay, and dumping out in those community adopted and supported file formats is good, because it makes the outputs of Maker immediately useful in the entire suite of GMOD tools like gBrowse, Apollo, Triple, Intermine, BioPearl, Gals, although we don't have those up here, but the idea. It's a good thing that we are all agree to speak the same language, and for genome annotation, GFF3 and FASTA files are the language for the most part. Okay, so that's through one section of the talk. I don't see any questions on chat that we need to stop for, so I think I'm going to just plow forward so that we have plenty of time for the tutorial part of Maker. All right, so the basic Maker workflow that I've just talked through is sort of conceptualized here with a two-strand Apollo view below. Basically, we put in the genome sequence. We do a little bit of work to prepare and initialize the data sets. We run repeat masker. Off to the side, we're running SNAP, Augustus, FGenes, H, GenMark, one or more of these ab initio gene predictors. We align our EST, mRNA, seq, whatever protein evidence that we have with BLAST. Those are filtered, clustered, polished by Exonerate, put back together, and then Maker, and here's the real power of Maker, is this synthesis step where it talks back and forth with these gene predictors and comes up with the annotation produced by those gene predictors that best fits the evidence. So that's a lot of computational work there, and if we did that all on our laptop and tried to annotate like the pine genome, we would finish in a few thousand years. So Maker has implemented a distributed workflow. I'm not going to go into detail about this because I know Carson will talk more about it later, but basically at many steps along the way, Maker will break up your data sets into smaller chunks, bite-sized chunks, and can run on large clusters a lot of data very quickly. That parallelization efficiency, this is just a simple chart to show that at least up to these 30 or 40 CPU cores that we're showing here, the more cores you give it, it linearly scales. And we've scaled it much past 40 cores, of course, and it really does scale quite nicely. In fact, Carson, earlier last year, and the paper came out earlier this year, Carson worked with the Pine annotation group to annotate the 30 gigabase, so we're talking 10 times the size of human genome in something like 37 hours on the Texas supercomputing cluster using 6,000 CPUs. So I believe at the time, and probably still today, that was one of the, certainly the largest annotation compute ever done, and one of the largest bioinformatics computes done in one job at the time. Okay, so there's lots of ways you can annotate genomes. So why are you going to choose Maker? We're going to go through a few things that I'm going to try to show you that Maker does a good job of annotating. But in addition to that, the title of this talk was Maker, the Genome Annotation Pipeline. These days, we really ought to add in there the annotation, maintenance, and management pipeline, because so many genomes, it's an ongoing process. You don't just produce one copy of your annotation set and then call it good. You have to maintain that annotation over time. So let's talk about a few use cases for Maker. So the standard, just a de novo annotation, and it provides quality metrics for you. We've just got an assembled genome, and we need to annotate it first pass. Maker is great for that. We see it happen over and over again. A genome will get annotated. Two different groups, genome will get sequenced and assembled. Two different groups are interested in annotating it, but they can't quite work together very well. And so they both end up annotating it, and then at some point downstream, either they or somebody else needs to work with both of those annotation sets, and they need to be merged together. And so Maker can be very helpful with that process as well. We're seeing all the time these days as next-gen technologies get better and better and sequencing gets cheaper, people get new evidence. Either they've annotated, they've sequenced another RNA-seq data set, assembled it into transcriptomes, and they want to add that in. Whatever new evidence comes along, you need to re-annotate with that new evidence. That new sequencing data could just as well have been genome sequence. So now you have more DNA sequence. You've made a new assembly. This assembly is better, but you don't want to lose all the work that the community has put into making and validating the annotations so far. So you need to map those annotations forward to a new assembly. Maker's your friend there again. And then, you know, at the end of all of this, it would do no good if we just made these annotation sets and shoved them into a database somewhere in text file format. Our community of users, the biologists who work on these organisms, have to be able to access this data in ways that they can use. That's what GMOD is all about, and all the tools for the rest of the week after this will be helping you share those with Maker, with your community, and Maker generates the file formats and its outputs in the file formats you need to do that. Okay, so let's talk a little bit about does Maker do a good job of producing annotations? And we'll ask this question in a few different ways, but to introduce that, to ask how well Maker's doing, we have to have some way of measuring how well Maker's doing. So one way in the genome annotation community that people have measured how well they're doing in terms of gene predictions is to run the annotation tools on regions of genomes that have been well annotated, where we have a gold standard set of genes, and then compare to that and see how well we did. Okay, so when we have that, if we make a gene annotation prediction that is exactly the same as the gold standard model, then we can say that we have perfect accuracy. So the sensitivity was 1. Everywhere that there was a gold standard nucleotide in this annotation, we have predicted it, and we have not predicted any areas as being part of that annotation that were not. So we've done very well. We can do perfectly accurate sensitivity, so we've captured all of the blue areas, but we've over-predicted, and so our specificity suffers, and so our annotation accuracy has suffered as well. Likewise, we can do poor sensitivity by only predicting a little bit, and we don't predict anything that shouldn't be predicted, but we haven't predicted everything that should be, and our accuracy suffers. Or we can do both, and we can get it wrong both ways. And these kinds of metrics, these sensitivity, specificity, and accuracy metrics, have been used for a long time in the genome annotation community to measure how well annotation sets do. So if we look at an example of how well Maker does compared to some of the ab initio gene predictors all by themselves, on some well-annotated genomes, Arabidopsis, Drosophila, C. elegans, and we look at these metrics, we can get some kind of an idea of how well Maker's doing. So let's highlight a couple of these, and we can see that Maker in this case has improved the overall accuracy and in this case we're looking at nucleotide accuracy, so at a nucleotide by nucleotide count, how accurate are our annotations. We can see that Augustus run by itself and then Augustus run within Maker, we've done a nice little bit of improvement here. I should describe this chart overall. These 3 columns here are the ab initio gene predictors, each Augustus, GenMark, and SNAP run by themselves. And these columns over here are those same gene predictors run inside of Maker, with Maker supervising them and providing the annotations. And then the 2 different numbers that we have for each of those are the accuracy at the nucleotide level. Did we get all of the nucleotides correct, or the accuracy at the exon level? Did we get exon boundaries correct? But we can also find examples. This is another example of Maker showing us a nice little improvement, and another nice little improvement on C. elegans. So on all these well-known genomes. But we can find a few examples out here where we actually looks like Augustus did better on its own than it did when supervised by Maker. So what's going on with that? Well, these genomes here are all very well annotated genomes that were sequenced many years ago, and in fact, they are the very genomes that these gene predictors were developed and built to be able to annotate. And so it's fairly well known in the annotation community that if you train a ab initio gene predictor well enough, then you will do an incredibly good job with the ab initio gene predictor at recapitulating those annotations. But it's really not so ab initio anymore when you've had to have perhaps tens of thousands of gold standard genes to train on. And in so many cases these days, the new genomes that are coming online are in lonely, isolated parts of the phylogenetic tree. And if you try to run ab initio gene predictors on those genomes, they can do fairly poorly. To simulate that here, we're going to lie to the ab initio gene predictors a little bit, and we're going to mix the HMMs around that we gave them, and this simulates a poorly trained HMM. And you may think that this is cheating a little bit, but we're doing it to make a point, and we'll come back to it later. So again, you can see that Maker is doing a nice improvement in its ability to improve the annotations. And even in the case before where it looked like Augustus had done better, even when the ab initio gene predictor continues to be given the wrong HMM, so it has the wrong HMM here, Maker is still able to provide hints back from reality, from the aligned evidence, that allow that annotation to improve. Okay, so I'm going to just, so that was examples of how we do, how we check how well we're doing when we have gold standard sets to compare to. But Maker doesn't have that as it's running on novel genomes, and we often don't have that for novel genomes to estimate how well Maker is done. So we're going to develop a proxy, and instead of accuracy, we're going to start referring to this as the annotation edit distance. So if we take that gold standard gene that we looked at in the last few slides, and then we think about what we're about to do with Maker, we're about to align some protein sequences, some ESTs, maybe some RNA-seq data. If we were to collapse all of that evidence down, we're going to, instead of a gold standard gene, our gold standard is now going to be that collapsed set of evidence. Okay? So if we revisit those annotations, we're going to see that gold standard evidence can now be compared to any particular of those annotations. So let's say that the green model comes from one gene predictor and the purple model comes from another gene predictor. Maker will look at that sensitivity and specificity and calculate that annotation edit distance. Now, in the previous example, we were talking about accuracy, and we were looking for values of 1 to be perfect accuracy. Notice and remember that this is a distance matrix, and so now low is good. Well, back up there. And so this means that there is a 0 edit distance between this green gene model and this blue one. So we have to make no changes at all to the green model to bring it into perfect alignment with our evidence. Whereas with these other gene models, we have to do some, and in some cases perhaps a fair bit of editing to bring them into alignment with the evidence. So in the case of AED, just remember that it's an edit distance, and so smaller is better. And these metrics were developed by the Sequence Ontology in collaboration with many of the Maker developers in that publication there. Okay, so we're going to see a number of graphs here coming up in the next little bit that use that AED metric as a measure of how well we've done. And so this is just a hypothetical graph to kind of orient ourselves to the ones that are about to come. Remember that a low AED is good, a high AED is bad. And so in these cumulative distribution graphs here, we're looking to see on the x-axis we have the AED value, on the y-axis the cumulative fraction of our annotations that have that AED value or lower. And so a very good annotation set would go up very quickly, so that most of the genes, maybe 70, 80, 90%, 95% have an AED value of 0.5 or lower. A very poorly annotated gene might have many annotations out here that had little supporting evidence, and so most of their genes fall in with an AED value approaching 1. Okay, the first example that I'm going to use to demonstrate how Maker has done, you may not be able to read this slide very well. That's okay. I'm just going to say that the TERR community has done a real nice job of developing a star system to describe how good a particular gene model is. The very best stars, 5 stars, would have all of the intron-exon junctions of a particular gene model fully supported by a single piece of evidence, whereas a very poorly supported, a single star, would have a little bit of evidence that there was some kind of expression data there maybe, but we've got no support for any of the splice sites like we do up higher, and not even all of our exons are covered, and in the worst case, we have experimental evidence only and no actual physical evidence for those models. Okay, so here's a graph of those AED curves again, and the point to show here that I want to make is that the red and orange are those 5 and 4 star annotation sets, and when we pass those through Maker and ask Maker to calculate an AED metric on it and then plot it, we can see that AED does a really nice job of recapitulating a good annotation set, and AED also does a good job of showing us those poorly supported, less supported annotation sets. Just one other way to look at that really quickly is to see that the AED metric recapitulates the efforts of manual annotation. So this is another one of those AED graphs with AED down here on the x-axis and the cumulative fraction on the y. We're looking at mouse from release 30, the mouse annotation set from NCBI from release 30 a number of years ago, and then the annotation set a few years after that. So 30 was one of the original releases of mouse annotation, and 37 was a few years after the community had been working with it. And we can see that these AED curves from the early mouse annotations reflect poor alignment between the annotations and the evidence, whereas those from several years later show a much higher alignment with the evidence. The dotted lines are simply showing the subset of RefSeq annotations that had NCBI's RefSeq's gold stamp of approval given at the NM accession number. And so the AED metric did a good job of capturing the efforts of the manual annotation community over those years of time. I'm going to just skip this section in the interest of time and move on. So let's use then AED to see how well Maker does. In these AED curve slides, we can see some TERR annotations from TERR 10, a representative set of those annotations. We can see a set of Maker annotations that are just done de novo. And then we can see a set of Maker annotations when we didn't allow Maker to add new annotations, but we just allowed it to update the existing annotation set. So here again, we see taking these well-supported TERR annotations that the community has been working on for many years and running Maker de novo in green, giving Maker no previous history of those well-annotated genes. Maker does a really nice job of recapitulating what the TERR community has worked very hard at. And over in this slide, we see the same thing for maize. All right. So here again, we've continued to work with data sets where there were, a history of community annotation. So even in this last slide, even though Maker didn't explicitly know about the annotations in that gold standard set when it did its de novo annotation, the gene predictors had HMMs that were able to train on a good solid set of gold standard TARE genes. So we don't have that luxury in many novel emerging genomes these days, and so we need a slightly different metric. And it's fairly well known that consistently across many well-annotated genome sets, about 2/3 of the genes, when we compare them back to PFAM domains and Go database, will be able to annotate some kind of functional category or some kind of a Go domain to about 2/3 of those genes. But for about 1/3 of the genes, we'll just know nothing about them at all. And so this is that kind of data collected for 6 reference proteomes, things like human, mouse, fly, worm, yeast, and so forth. And if we look at a similar kind of a curve for a brand new genome that was just been annotated in the last few years, this is one of the ants that have recently been annotated with Maker, and then Schmittea mediterranea is the planaria that was annotated, one of the first genomes annotated with Maker. If we run the gene predictor SNAP all by itself, as we're seeing in this column here, we can see that of all of the annotations that SNAP produced, only about 6 to maybe 15% of those annotations actually had some kind of a known functional domain within them. And almost 90% or more of the genes predicted just did not have any kind of recognizable protein domain. When we run those same novel genomes with SNAP supervised by Maker, we can see that Maker improves on that greatly. We may still not be all the way up to what the well-annotated genomes look like, but if you have to hand off a starting set of annotations to your community, you'd much rather hand them something like this, where the majority of the gene models are going to have some kind of a protein-looking domain, rather than sending them an annotation like this. Okay, so I think given that I've taken up pretty much an hour here, we got a little bit of a late start. This last section here talks a little bit about the Maker web annotation service. I'm going to skip that entirely. It's talking a little bit about how do you get Maker on the website, some of Maker's publications, a little bit about Maker installation. Basically, it's pretty easy to install these days. It'll install all of the Perl prerequisites for you. It installs the necessary executables. It's quite a challenge to build an install script that can install all those kinds of dependencies for you. But you know, Carson has worked very diligently to build a good install script and keep it up to date. So we have a lot to be thankful for there. Maker will even install MWAS or the MPI libraries for you if you ask it to. So these are all things that Carson will be covering next, and I don't want to take up more time from him. Maker has over 30 accessory scripts. A number of them deal with converting to and from the various file formats for authors of tools who were not as thoughtful as Maker has been in talking a standardized format. Many of these tools have just invented their own format, and so Maker provides conversion scripts for those. Maker provides a number of scripts that help to add those functional annotations that I talked about earlier, and I know Carson is going to cover that. And Maker provides a number of scripts that help you map IDs and models and data sets forward from one assembly to the next, although much of this is handled directly within Maker, and you don't even need those scripts anymore, but they're still there if you've gotten used to using them. Okay, so then just finally in closing, all of this is work that comes out of Mark Yandel's group at the University of Utah. Come swing by and see us on the web sometime. A lot of the work is built on the standardization, formatting, and ontologies that are built and maintained by Karen Eilbeck's group in Department of Biomedical Informatics, also at the University of Utah. So a real shout out to them for helping bring sanity to this field of genome annotation. Acknowledgements. I acknowledged at the beginning, but I have to reiterate the importance of these three gentlemen: Carson Holt, the primary developer of Maker, Mike Campbell, and Daniel Entz are taking over most of the annotation efforts in the ENDL lab these days, and Mike has also started to take over some of the development efforts. So, and I believe that all of three of them are online this afternoon answering questions. And then Karen Eilbeck's group in the sequence ontology, the rest of the Yandell lab, and a number of professors here from Michigan State University who are co-PIs on the current Maker funding. And I should have had something in there, but I think I ended up deleting that slide. But thanks to the NIAH, no, actually the current Maker funding is NSF, so thanks a lot to the NSF for current funding on this project. Okay, I'm going to stop there, hand the reins back over to Carson. We can unmute all for just a second. I've seen you open by just a second. I've seen some by.
Okay.
Okay, I've unmuted everyone in case there's any.
I've unmuted everyone in case there's any. So I'm going to just say one more thing since I was just leading out here. We really want to move forward quickly for Carson's section. I suppose if, Carson, do you think we want to just take a really quick 5-minute break for people to get a cup of coffee or something, or shall we just?
I think we should take a 5-minute break.
All right, let's take a 5-minute break, and we can then the instructors can look back through the questions here and try to make sure we haven't missed anything while we do. Thanks, everybody.
Okay, I imagine, can everyone hear me? I have no indication on my screen. Okay, perfect. Everyone else should be muted. So let's go ahead and go to the online schedule. If you click down here on Maker, it will automatically take you over to the wiki page. I'm going to go ahead and just cut and paste this link into the chat window in case someone needs to click on it there. All right. So Barry basically covered a lot of the theory behind Maker. So we're going to skip down to section 6, which is talking about Maker installation and then a few examples of running Maker. So to begin, we're going to go ahead and set up launch another AMI, just like we did with SOBA. So you need to log on to the EC2 management console. Here I am logged in. And go back to the tutorial. You can see here we need to search for AMI 28E70E40. So I'm going to go ahead and go to public images and search for it. Now, this is the Maker 2.315 tutorial version 2 is what you should see here. And we're going to go ahead and click Launch. Now, you should run this on at least a medium instance. I'm going to run an actually much bigger instance because we want to show off some features that you don't necessarily need to run that have to do with MPI. Okay. So looks like we have someone falling behind. So the first thing we need to do, like I said, is go to the EC2 management console like we did with SOBA. Make a short link so you can. Okay. So I'll just go ahead and paste the link into the chat. So go ahead and click on that link if you need to get into the Amazon Web Service. Then you go to you sign into your account, and then you go to AWS Management Console right here. Then EC2. Back up here on this window. Now we're going to click on AMIs. And these are my AMIs. But if you also are following along in the wiki, which is the other link in the chat, you will see that here in section 6, I have you search for an AMI named AMI 28E70E40. So I'm just going to copy that value. Or you can search for Maker _231.5. That also works too. So back to the management. Click here. You need to be in the US East region to find this. So up here in the right-hand corner, I select US East. Then here I select public images, and I put the image I'm searching for. Now, if we right-click on the AMI, you can go to Launch. Like I said, for our purposes, you should at least launch a medium instance. I'm going to be launching a larger instance to show off some features of MPI, but you should be able to follow along with everything I'm doing using the M3 medium instance. Go ahead and click Next Configuration. Nothing needs to be changed here. Nothing needs to be changed here in this section, so you're going to go ahead and go forward. Give it a name. I'll call this Maker Tutorial. And finally, you don't really need to change anything here. I'm just going to add HTTP just case. And then you hit review and launch. Just wait a moment, make sure everybody's caught up to me here. So once you hit review and launch, you go to this screen, then we'll hit launch. And you can either create a new key pair, or you can choose an existing one. If you choose an existing one, you type in a name for your pair, whatever you want it to be, and click download pair. It might download as a text file or a PEM file. The extension could be either one. You just need to make sure you know where it gets saved to. I'm going to choose existing pair. Here we have GMOD pair, which I created earlier, and we can use to log into Barry's AMI. You can use the same pair that you used to log into the previous MAI for the SOAP example. You have to click I acknowledge that you have access to this private key. And then you, of course, launch your instance. All right. Once your instance is launched, you can go back and look at your instances, and you can see this is still pending, and it'll take a moment for it to launch. Okay, once the instance is running, it's still running some status checks, so you might not necessarily be able to connect, but at least you can get the connect information. So you click on the instance and you click on connect. This will bring up a window of the information on how to connect to this instance. If you created a new key pair file, you need to make sure you open a terminal and run this command on your key pair file. Otherwise, it will fail to connect to your instance because the permissions are too lax. It wants it to be a file that's only available to you and not available to the rest of the world. And then you can use this string here below to connect to your instance. So I'm going to go ahead and open up a terminal window. You can see, is that big enough for everyone? I can zoom in if needed. Make it a little bigger. All right, so we have SSH. I have my GMOD pair file is in my downloads. And you can see that when I downloaded it, it had a .txt extension. You can either rename it to remove that extension, or you can just leave it as is. As long as you modified the permissions, you're fine. All right, so you can see here, I'm going to connect to my instance. If it's up and running, I should be able to connect to it. Oops, saying I need to change my username. So let's change that. Instead of root, it needs to be EC2 user. Right there. You can also see that it should say the same thing on the wiki page. Here we are. It says SSH-I, your private key, and EC2 user at whatever the address is for your instance. Okay, I'm going to go ahead and connect here. And now I'm up and running. I'll give everyone a moment. Is there anybody, because we'd like to have everybody with their instances running and be able to follow along the examples. if you're having problems connecting, go ahead and just paste it to the chat. Like to make sure everybody's able to keep up. Remember, you do need to, if you've downloaded your key pair. Okay. I'm seeing an error here. Okay. For this error, what you need to do is you just need to, you can either edit your known hosts or you can delete the file completely. The offending entry is this one here. What you can do is if you open your known host file, you can search for this entry, the EC254 compute AmazonWS.com, and then just delete the entire line, and then save the file. You need to use a text editor like Emacs or Nano. Okay, so this is back to the management console. So if you have already launched the instance, like I said, your instance will appear here. You can click on instances to see all of your instances. And this was the one I actually just barely launched to make a tutorial. Yeah, Scott's right. You might just be easier to delete the known host file. Just give you an example of the command. And then you can try reconnecting with SSH. Okay, and once you have your instance, if your instance is launched, this should say running, and you'll have your status checks all 2 of 2 checks will have completed. And if you select your instance, you can hit connect to get the information on connecting to that instance. So to make your key private, chmod 0400, and then the key's name is the command you would use. I've pasted into the comments. Okay. And you can just cut and paste this. Instead of root, it needs to be EC2 -user. If you put this command in, it'll actually give you an error telling you which user to switch to. All right. So I'm just going to log out of my instance again so you can see how I logged in. SSH-I, this is my key pair. If the permissions are incorrect, chmod 0400, the location of your key pair, and that will fix the permissions. And make sure you have your user as EC2-user, and go ahead and SSH. All right. I think most people by now have been able to log in. There's still a couple of people who might be having issues. Of course, Scott and Barry will be working with you to try and make sure you get connected. Right, if your permissions are denied, make sure you have the right username. You have EC2-user. If you can also use, for example, here, there's a public IP address. If you don't want to use the long name here, you can replace this entire section with this IP address. Not the one I'm using, but you can copy whatever's on your instance. You can see copy. Now, go ahead and log out, and I'll show you how that works. And you can see I'm still ECD user. Instead, I'm using the public IP address, and that works as well. Okay. If any users who've still been unable to connect, go ahead and just let us know via chat. I'd like to know how many people I'm still waiting on, and whether it's probably time to just move forward while Barry and Scott help figure out what the connection issue is. Not yet? Okay, we've got one person not connected yet, 2. 3. So the permissions and the key, chmod 0400 will set the permissions. Way up. 0400. That command will set the offer permission. And if it's saying public key, you also might need to have your username incorrect to make sure that that's right with the connection string. Right, so you once again, and here's where I change. I can use either of these values: public IP address, which you can find on, or the other address from your connection string, which is in the console. Click on it, connect. This gives you the full string here. Or you can use the public IP address to connect, which would look like that. The username depends on which AMI you're using. If it's a Ubuntu Linux, it's usually a Ubuntu. EC2 user is the Amazon Linux default user, and that's why it's EC2 user, because it's the Linux kernel used here is Amazon Linux. Okay. Once you're able to log in, you should be in your home directory. And you will have the Maker tutorial here in your home directory, and that contains the examples we're going to be going through. But before we do that, I'm going to describe a little about the Maker installation itself. That way, people who haven't connected yet can still work in connecting while I describe how Maker is installed. So Maker has several prerequisites. Let me go ahead and make this page bigger. So you can see there is a number of Perl modules that you need to have installed. Maker also relies on another number of external programs like Snap, the Gene Predictor, Repeat Masker, Exonerate, and Blast. And optionally, you can have more gene predictors like Augustus or GenMark or FGenes-H. And the Maker package can be downloaded from http://yandelab.org if you want to install it locally in your laboratory. Here, once again, this is how you connect to your instance, the SSHI, with this information coming from your connection string, from the console. And once we are on the instance, everything is actually installed for you, so you won't have to connect. So let's take a look at the actual Maker installation. So let me make this bigger for you all. Okay, so if we go to user local, we can make it even bigger. Okay, so cd user local, then Maker. And then if you do ls-1, you can see all the contents of the file, or ls-l if you want more information, or just ll, which is what I'll probably be using most of the time. The tutorial says ls-1, which is the simplest way to see the output. But you can see there's just a number of directories here. The most important file here when you're setting up Maker is the install file. Take a look at that. So with less install, you can see that there is an easy install information which has just 4 steps for installing Maker. We try and make it easy as possible. Maker will automatically download any dependencies you give it. For example, you can run build.install deps, will install missing Perl dependencies, or build install exes will install missing executables like Snap or Exonerate for you. I'll show you an example of that in just a moment. You can also install Maker with MPI, either OpenMPI or MPI-CH2. And later down here in the file, you'll see there's a detailed install. If there's any errors that happen in the easy install, then you want to read the section on detailed install. But we won't actually cover that right now. So when you install Maker, you go into the source directory. You run perl build.pl, and this is where you tell it your configurations. You don't have to run it. I've already installed this for you. So you can just watch me. Perl build.pl, it'll ask me a few questions. Do I want to use MPI? The default answer is no. And then you can see it says all my dependencies have been found. Everything is working just fine. If I was missing a dependency, say Exonerate, I'm just going to remove this just to show you what would happen. Then when I run this perl build.pl step, you can see it says I'm missing Exonerate and I need to install it. I can either run build install exes to install all missing dependencies, or I can go ahead and let Maker just install it by build Exonerate. And it takes just a second. It reaches out online, downloads it for me, and will install it where Maker knows how to use it. Now, if I were to run this again, you would see that all my dependencies are now satisfied, and build install will install Maker. If I decide I wanted to use MPI, let's go ahead and scroll down in the wiki page. You can see MPI gives you a lot greater throughput, because you can imagine this is gene annotation can be very computationally intensive. So having more CPUs at your disposal over a computer cluster can make it much, much more faster. And as you can see, as you use more CPUs, it scales virtually linearly with greater throughput with more clusters, with more CPUs. If you want to install it with OpenMPI, you have to export a command I explained here. to pre-export shared libraries for OpenMPI, you have to put that in your bash profile. I won't show you how to do that today, but it's in the installation instructions. And if you want to use MPICH2, you just have to make sure you compiled MPICH2 with enabled shared libs flag set. Otherwise, it won't have shared libraries for Perl to access. Here's an example of installing with MPI. Quickly, Perl build.pl. And I'm going to say yes to this question. You don't have to follow along if it's already installed for you. You can see, and then I have to tell it where MPI.h is located. And it is located in right here. User includes OpenMPI x64. And once I do that, Maker is configured with MPI. I do build install. And Maker can either be run with or without MPI. There will be a couple examples where I'll run it with MPI just to show you how it works. But really, all the examples you don't need MPI to run here. I prefer OpenMPI over MPICH2 if you have large clusters. It scales better. You can get several thousand CPUs in OpenMPI. With MPICH2, you're probably limited to about 100 CPUs per job. Okay. So now that we have Maker, I've shown you how Maker is set up and how it's able to install its own dependencies and a little information about MPI setup. Let's go ahead and go to the Maker tutorial. It's in your home directory under Maker tutorial. You can see here there are 4 example folders, examples 1, 2, 3, and 4. We also have these here in the wiki. Okay, let's do skipping a step. Now, after you've installed Maker, I guess one of the most important things is to make sure it's working, so you can just run the help statement, and this will provide you the usage statement for Maker. As you can see here, we have version 2.3.1 installed. Maker takes a series of control files, ops, bops, and exe. They basically tell Maker all the options for your genome annotation. I'll explain them in more detail later. Maker says several command line options. For the most part, command line options are just used to override options that are already in your control files. So you don't have to use these if you've already set them in your control files. One nice command line option is the -quiet. Maker produces a lot of output. So it's nice to, if you are doing a large genome, you can silence that output. And Maker-CTL, this is an important one. This generates empty control files. And with those, I don't know how to make my mouse highlight to better use it as a pointer. But you'd use Maker.CTL to run these, to create empty control files. So that's probably the command you'll use most of the time. The rest of these can be set directly within the control files themselves. All right. So here we have our 4 directories, examples 1, 2, 3, and 4. Let's go ahead and look inside example 1. CD example 1. And inside here we see you have a number of control files, a number of FASTA files that we're going to use for our example job. These files actually come from through local Maker data. When you first download Maker, it comes with a number of example files just to make sure the algorithm is working. These 3 are an example annotation job for Drosophila. It's just a single gene. This is an example annotation job for human. It's just a handful of, I believe, 5 genes that you can set this up as your genome assembly. This is your EST evidence. This is your protein, and just run. So let's go ahead and set this example job up. The other files here we have is finished.tgz and partial.tgz. If you were to fall behind at all, you can actually untar this, and it will have the final results already in them, so you can skip ahead. And if you are, and this file is just a copy of some options we're going to set, so if you fall behind, you can see what options I've set either in the wiki or in this file. And this one is just a partially complete file. Maker can tend to run for a long time, so on a few of these examples, I'll have you open up this partial TGZ file to pre-process. It'll dump some pre-processed data out for you, so that Maker will just run faster. But don't worry about it. I'll show you more in just a minute. Okay, so I hope you're all following along here in the wiki. Now, so Maker's primary input is FASTA sequence. If we look at the FASTA file here from the Maker data, or you can either use the DTP protein file that's in this directory as well. You can see that it's just a series of FASTA sequences for protein data. Here we have the contig. This is about a 100 kb segment of Drosophila chromosome. And to get started, the first thing we're going to run is maker-ctl. What this is going to do is just going to generate some empty control files that we will use to run Maker. So maker-C, capital T, capital L. This created 3 files, maker bops, maker exe, and maker ops. Let's take a look at these files individually. Maker exe just contains the location of various executables. You can see here we have a location of blast, repeat master, exonerate, snap. So if Maker tries to automatically detect these and can't find them, you can manually fill out this file with the location of these different programs. For example, if you have fgenes H in a private folder, you can tell Maker where it is, and Maker can use it. The next file is Maker bops. This means blast options. You can see here that by default, Maker is going to use NCBI BLAST plus. If you have different flavors of blast, like standard NCBI or blue blast, you can also set those here. These are just filtering thresholds that Maker is going to be using to filter genes. You can see percent coverage blast N means in order to keep evidence, I need at least 80% coverage end to end. I need to have a percent identity of close to 85%. The E value given by BLAST must be at least 1 e to the minus 10th, and this is the bit score per HSP. And it's good to set this high for BLAST N results because these are ESTs. We expect them to be nearly identical. And you can see here with BLAST X, which is going to be protein evidence finding against the genome, we have much more lax thresholds because of distant protein relationships. We expect to have less perfect alignment. And T-BLAST X is for recovering ESTs from another species aligned back against the same species. But still, we use a high threshold looking for near complete identity. And these, of course, this is just for transposable element alignments for repeat masking and exonerate alignment thresholds. Now, the main section of Maker is the maker ops.ctl file. If you look into this file, you'll see several sections. We have the genome section where we identify the genome assembly and what type of organism we are annotating. And Maker supports both eukaryotic and prokaryotic organisms. It just needs to be told what kind of organism it's working with. The re-annotation section you can ignore for the examples we're working on now. This is if you have an old Maker job that you want to pass through with new evidence. This is where you put your EST evidence. EST equals, we'll put a FASTA file location here. Or alt EST, this would be ESTs from another species. You can put a FASTA file here. Or if you have a GFF3-based files that already have aligned ESTs, this might be things from cup links for example, where it's already aligned the mRNA seq reads to your genome, and you just want to pass in a GFF3 file, you can provide it here. Protein evidence, and here we select repeat masking options. By default, it uses the entire rep-based library. A model organism equals all. You can use specific species-specific libraries already built into rep-based by defining different organism names. The repeat masker documentation defines which organism names you can use. Or if you've built your own species-specific library, you can provide it to rmlib equals. This would be a FASTA file, something that you would have gotten from programs like Repeat Modeler. This is the default repeat protein library that comes with Maker. And here down below, this is where we set what gene predictors we're going to be using. I can provide HMM files for SNAP, GenMark, a species file for Augustus, or FGenes-H. I can also do what's called EST to genome, where if I set this to 1, I will align ESTs directly to the genome and try and infer the model without an HMM, just using direct alignment, or protein to genome, where I use protein alignment to try and infer gene location. This is good for training because it can generate rough models that can then be used as to seed the training of ab initio gene predictors. Recently, we added the option to run tRNA scan. If you set this to 1, it will look for tRNAs in your genome. And the remaining options are mostly thresholds and behavior options. For example, you can identify whether or not you want to consider single exon as evidence, single exon alignments, or whether you want to not allow proteins below a certain length. For example, partial protein calls may be very short and you want to exclude those. You can set a minimum protein length. Or right here, split hit, this is the expected maximum intron size. So if you want to keep alignments from jumping across large distances, you can make this threshold lower for example, fungi or oomycetes or other organisms that have high gene density and very short introns. You can lower this number to restrict evidence alignment. Okay. So the next step is we're going to go ahead and edit this Maker options file to use the files that we want to give it. So you can either use Nano, Emacs, or the program you wish. And we're going to set the genome equals option to the DPP config FASTA, EST equals, and protein equals. And we're also going to set EST equals to 1. So we can use the ESTs directly to create genes. If you fall behind, you can also just copy the options file directly to makerops.ctl, as this file already has these options set for you. So I'll go ahead and open this. I'll use Nano just as the documentation says, because it's a little bit easier for most people. So dpp config.fasta is the name of the file with an underscore. Here we have est equals dpp est.fasta and the protein file dpp protein.fasta. And we're going to set est equals 1. And just so you can see that for a moment. Protein homology evidence, just to answer your question, is very easy to get. You don't have to be from the same species. You can use, for example, the UniProt SwissProt database is a comprehensive database, or you can pick the entire proteome of a related species and then include that as your protein data. And in general, DST data is sufficient to get at least training going, but when you do your final annotations, you should include the protein data. Let's see, is it EST or proteins? Just to check the file names, protein without an S. Okay. Then we want to save this file. This is Control-O. Enter. So right here below it says write out, Control-O, And then here we see down here below, Control X is exit. Control X, and now we're out. All right, so we've created this file. If you're having trouble editing it, you can also just copy this file, cp ops.maker to maker ops. Okay, now go ahead and run this. Just type maker. Yeah, maker, you can either give these files one at a time to maker, or if maker finds it in the current working directory, it just knows what the files are supposed to be called. So I hit maker, and it will start working. You can see now it's processing control files. You must have, if it says your file doesn't exist, it's because you wrote the name incorrectly, most likely. You might have an S at the end. DPP_EST should be the name of the file instead of DPP transcripts. Oh yes, that's an edit in the wiki. It should have been EST right here. So Barry can edit that. There we are. So now it should be fixed if you refresh your wiki page, DPP-EST. All right. Now you can see Maker is performing a number of steps here. At first it performs repeat masking using repeat masker. Then it goes on to align things using blast and exonerate. And it ran relatively quickly. It says 63 seconds went by, and we have a finished job. So Maker is working correctly, and we should have some initial Maker output. So I'm going to skip this section here describing what we just did, because Barry pretty well described the steps of repeat masking, gene prediction, etc. And we're going to skip right down here to Maker output. So Maker writes its output into a directory directly named after the input assembly. So it's called dpp-config-maker.output. We're going to go ahead and jump into that directory, cd dpp-config.maker.output. Now, if we look inside that directory, you can see there's a number of other files. This is a log file of the options that were used to run our Maker job. This is a directory containing just the raw data, the error-corrected databases that protein evidence, ESTs, etc., that were used for the Maker job. And this file is probably the most important, this dpp-config-maker data store index. Let's go ahead and look at this file. You see it contains two entries. The name of the config, the location where data for that config is stored, and its status. It says we started running it and then we finished. There are other statuses available here in the wiki, so you can see failed, something went wrong, retry, something went wrong and it's trying again. Skipped small, that means that it was too short to annotate. Or died skipped, that means after a certain number of retries, it decided not to retry again, and it's just going to fail. If you're having problems with Maker not running because of failures, copy this file to maker.ops, just to make sure you have everything correctly. Ops to maker ops.ctl. You might just have a text error in your input, and that will put the options the way they're supposed to be before you can run Maker, before running Maker. Now, the most important thing about this file right here is the location of where the data for each config is stored. Maker actually stores every config or chromosome separately. So if we look inside that directory that was given to us in that file, I need to be in this directory, sorry. So I'm inside this directory, and if we look inside dpp-config data store, you'll see there's a number of files just for that config. We have a GFF3 output for that config. Maker produced protein FASTA, transcripts, a run log just telling Maker internally what failed and what didn't, and the void. For the most part, you can just avoid this. You don't have to know what's in there. It's raw data files from blast, exonerate, etc. So if you ever rerun Maker, Maker can automatically look here to see if the file already exists, before trying to rerun analysis. Let's go ahead and look at this file. Oops. Let's go ahead and change to this directory. I'm going to back out. I'm going to add a -S to my left command line. Reason being that adding -S causes large, long lines to wrap. Okay, for those of you who Maker just finished, let's go back to here, CD Maker tutorial. Example 1, basic. And you can see here, this is the output directory. dppconfig maker.output is where Maker produces its output. And in here, there will be a file. I'm going to go to that directory called dppconfig master data store index log. And in that file, it tells me that my job finished, at least for this config, and I can also copy this directory. That directory should also be indicated in here in the wiki. And we do cd data store to that directory. And when we look inside, we'll see the results just for that config. Once again, less -S for make it wrap around. And we can see we have a single gene in this file, as well as other output. We have repeat masker data, blast in results, EST to genome results, protein genome, and then the sequence of the config we were just annotating down here at the bottom. And you can see here there's rather long attributes. We have the gene name here, this ugly name, maker config DPP 500 exonerate EST to genome gene 0. In this instance, we have 3 mRNAs produced because it was EST to genome data, and the EST suggested 3 different isoforms, and those are in the file. We also have the output proteins. You can see here, this is what the proteins for each isoform look like, mRNA 1, 2, and 3, and the transcripts FASTA. This is isoform 1, 2, and 3. Okay. Now, the GFF3 files, they're good for, the config-based GFF3 files are good for programs like Apollo, which can only look at one config at a time. But other programs might be able to look at multiple configs at a time. You give it a GFF3 containing all of your configs. I'll show you in just a minute how to merge, create and merge GFF3 files if you were to give an input assembly that had multiple configs. So, to do that, there are 2 accessory scripts that come with Maker. There's GFF3 merge and FASTA merge. So, I'm going to go back to Maker tutorial, example 1, basics, the directory that we were running in, and back into my output directory. Now, if this data store contains hundreds or even thousands of chromosomes or contigs, then we want to create a merged file, we can use GFF3 merge. And you see this GFF3 merge can take, with a -D option, takes this data store index file that I talked about earlier that tells it the location of every config and whether or not it's finished. So if we do GFF3-D, DPP config master data store index log, which you can also see here. This only takes a second to run. It creates a file, dppconfig.all.gff3, which would be the merger of all the GFF3 files. In this case, there's only a single config in the output, so they all get merged into that one file. To do the same thing with FASTA merge, cut and paste this command. Fast emerge, just to show you what the command line is like. You can see, once again, it takes this -d data stored index log, or you can give it a list of multiple FASTA files. But it's easier just to give it this. And it creates a merged file for all of the contigs, dpp config.all maker proteins and all maker transcripts. And then you can use these as these would be your final genome-wide files, while the data store contains individual config files. Okay. The next thing we want to do is, well, how do you view these annotations? This can be done using, well, we can look at it on the command line using less, as we showed you before. We can use this -s dpp config all. Oops. And you can see here, this is the contents of the file. But GFF3 output by itself, it really isn't that useful. It's not that nice to look at. We want more of a visual representation. So for that, we have programs through GMOD. There's Web Apollo, gbrowse, and jbrowse. jbrowse is a very convenient way to do it. In fact, jbrowse comes with a script called maker2jbrowse. Maker2jbrowse, basically you just go inside the jbrowse directory, call that script on a maker GFF3 file, and it will populate the GFF3, the tables, the JSON data for jbrowse automatically using the maker file. It's extremely simple. I've already done this for you, just so you can see what this file looks like. If you click on this example, this is what that same GFF3 file we just produced would look like in jbrowse. So once again, what I did is here on the wiki page, I'm going to click on this link, which is just going to send me to a jbrowse instance where I've already loaded this file. I'm going to leave how to load those files as an exercise for the jbrowse files. But if you remember before, we had 3 isoforms. So we have 3, 1, 2, 3, and you might have to turn these on or off depending on whether or not they're automatically appearing in your viewer. But you can see 3 isoforms from Maker. It displays the full names for the genes. These are the EST evidence, and this is the protein evidence supporting these different isoforms. And you can see that the ESTs, because they are different enough to where you can tell that they are not from the same isoform, and Maker can recognize the difference using the EST to genome option to try and recapture some of the isoforms data to predict multiple transcripts. All right, and you can see here basically the exons and introns align across the different evidence types, suggesting that that Maker did a relatively good job. If we were to click on one of these, we can look at some different values. We can see here the AED, annotation edit distance score, for different isoforms. This is how well the isoform matches the evidence, or the transcript matches the evidence. You can see a 0.14 value. A value of 0 means perfect match. A value of 1 means perfect mismatch with the evidence. So lower values are generally better. And if we were to process the whole genome, we could sort genes based on their AED values to isolate the genes that had the most mismatch with the evidence and focus our annotation efforts on those. And we'll actually have an example where we're looking a little bit at how AED can be used to merge legacy annotations. So, once again, this is here in jbrowse. This is just where I've already loaded this into a separate server. I click on this, and it will bring up this window. You might have to zoom out. You also, here on the left or right, I believe you do have to have Java script at least enabled to be able to view jbrowse. And this isn't on the instance. This is actually on a global web server. So this is it should be accessible to anyone in the world. So you were actually just accessing a computer here in Utah. if you were to click on that link. Okay, this error you're getting about the rep base, you might have started too small of an instance. That's an out-of-memory error for Repeat Master. Did you start at least a medium instance? I know if you start the micro instance, Repeat Master will fail. But that's exactly what this, what that error is from. You can also, if you're having a problem following along with that, let's go back one directory. This is what I wanted to show you before this partial. Let's say if I were to completely obliterate my output, I'm going to, okay, I just tar-the, you don't have to put the V there, ZXF will untar that directory. And what it does is it's going to pre-fill this with reports so that when Maker runs, it doesn't have to regenerate the data. So if you're having a memory issue in your machine, you might want to run this. It'll at least get you past the Repeat Master error because it will pre-fill out the Repeat Master report. And then I just type Maker, oops, and you can see it runs just like before. But I'm going to scroll up so you can see it says rereading exonerate report, rereading Repeat Master report. So Maker is smart enough to know that those reports are already finished for it, and it just doesn't bother rerunning Repeat Master, just pulls out the data. So you can see how Maker would run without having Maker run Repeat Master, which is killing the memory. And if you want to, you can actually, if you use that untar that partial directly in all of the examples, you won't even have to switch back out of the micro instance. It's okay because you'll still be able to follow along with the tutorial. You just have to pre-fill out the reports using that partial .tg file before running Maker, and then it will still run even on the micro instance. Okay, so let's go back. So now we've seen how Maker output looks in this JBrowse example. Very, very simple single gene example. The next thing we're going to do is run a little bit more advanced option with Maker. When you're annotating a new genome, the first step you need to do is train an ab initio gene predictor. And to train an ab initio gene predictor, you need genes to train it with. They kind of need to know what the genes look like in order to predict the genes. So Maker provides a way for you to produce basic partial or at least incomplete models that are at least sufficient enough to seed ab initio gene predictor training. And we're going to go ahead and show you how to do that. That way you can jump right into a new annotation project and go from there. So let's go ahead and move into this. Okay, this is configuration in detail. Once again, this is the genome options which I prescribed previously, re-annotation, EST evidence, protein homology, gene prediction options. But let's move right forward to the training ab initio gene predictors. Okay, so let's go ahead and change to this directory. Now for this, I'm going to actually do something for you. Make this nice and big. So I'm going to run this example on two different instances just to show you what the options of Maker running on MPI as opposed to just running linearly. a quick comparison. So let's go to Maker Tutorial, which is in your home directory. And we're going to go to example 2. Here we are in example 2. I'm also going to do the same on this other instance. All right. Inside example 2, you'll find these files. This is just the examples I'm going to be running that finished for you. This is step 1, finding genes. Step 2, running your newly trained predictor. And 3 is bootstrap training of the predictor. These are the options. In case you fall behind, you can copy these, your options, straight to your options file. And this is what I called about the partial file, where you can pre-fill out Maker results to make Maker run faster, or use less memory if you're on one of the micro instances. And this is the input file. We're going to use an input genome config. It's about 1.7 megabases in length. It has to be pretty big because we have to have enough genes to train with, which normally means Maker's going to run slow. So I'm going to have everyone actually at least pre-fill out some of the data to make Maker run in a normal time. Here's the EST data, and here's some protein data we're going to use from multiple species. This is from a genome called Pythium ultimum. It's an oomycetes. It's genes. It's not a fungi, but its gene structure is very similar to fungi. Very long exons, short introns, and high gene density. So even in a very short 2 megabase region, we're going to have a lot of genes. Okay, so we've looked at the files in the directory. Next step is to run maker-ctl to create the control files and then edit the Maker options file, or you can just copy the options file directly. Let's go ahead and do this. So maker-ctl. Now I have my Maker files. I'm going to go ahead and edit this using nano. Okay, the genome file is going to be pyuconfig.fasta. The EST is pyuest.fasta. That is down here below. The protein is the species protein FASTA file. And we're going to set EST genome to 1 and protein genome to 1. What that's going to do is it's going to produce gene predictions directly from ESTs and proteins. I'm going to go ahead and save that file. Yes, we want to save it. And to get out, there we go. So once again, pyuconfig.fasta in the genome, EST file, protein file. It's best to just copy these values right out of the wiki page here. And I'll give everyone a moment to set those. Now, while you're setting those, I'm going to do this, but you don't need to. I'm just going to run it for a second just to show you what it's like. Okay, and this one, this is my other instance of the low. This is a medium instance, and this is a large instance. So I'm going to show you some examples on both of them. So the options are here on the wiki page. Genome equals pyuconfig.fasta. EST equals pyuest.fasta. Protein, sp protein.fasta. EST to genome equals 1, and protein to genome equals 1. Yes, for most genome annotations, you never actually have to edit these other two files, the blast options of the exe files. This is only necessary if you need to show the location of non-standard executables, and this is only if you want to change blast filters, but you really don't need to. So this is the only file anyone ends up ever editing. I'm just, on this one, just to make it fast, I'm just going to copy this straight to here. That will fill out the data. Okay. So now I have two instances ready to run, an extra large instance and a medium instance. Okay, I'm just going to, you don't need to run this. I'm just going to run it for just a second so you can see the difference. I'm going to type maker, and this is the medium instance. takes a moment to process the facet, and then you can see right away it starts processing repeat masking. This gene config is about 1.7 megabases in length, pretty long, and you can see it's taking a little while for each step. It's going to run BLAST 10 times, and it's run repeat mask, and it's going to do several other steps before it even makes it to the next section of the config. So about now, this has processed almost 1 KB. If I let this run to the end, it will take about 20 minutes, which I don't want everyone to wait for. Just wanted to show you how slow that was, but just to show you how it works. So instead of having everyone do that, we need to obliterate the output, and we're going to we're going to untar this partial TGZ file, and this is going to give us some pre-computed output that will run much faster. Otherwise, we'll have to sit here for maybe half an hour. Okay, so I was just showing you an example of how slow Maker was going to be on that config. It took about 40 seconds for it to go to about halfway through 100 KB of sequence processing, and there is about 2 megabases to go through. So we would be looking for at about 20 minutes of processing, which we don't want to do. So I pre-filled out this partial output for you by untarring this partial.tgz. And I explained that here in the wiki as well. tar-zxf partial.tgz. And this is just going to pre-fill out some, so it won't have to rerun repeat masker, won't have to rerun BLAST, because alignments take a long time, especially on a config this size. But I need a big config to get enough genes to train with. So once we've untarred this partial.tgz, I'm going to run Maker again, but I'm using this base option, telling it to use the partial output in this directory. So it's going to try and run inside this directory, config1.maker.output, because I gave it base, config1. And I'm going to go ahead and hit enter. And instead of running the individual steps one at a time that takes a long time, you'll see it's just rereading our already processed reports. So this will go much, much faster. So it'll be done in about a minute. This is the large instance. I'm going to do the same thing, but what I wanted to show you here was the example of how to run it via MPI. Here I'm going to use 32 CPUs. You don't need to do this because you don't have a large instance running this. And if I were to run Maker on 32, we can compare how long it takes to fully process this compared to the medium instance, which is using pre-computed data. So the pre-computed data has a serious head start on us. Maker works. Okay, Maker works. MPI exec to Maker. It sometimes takes a little to start up because it's creating a communication ring. Okay, so this ran in 75 seconds because it was using pre-computed data. And once it's finished, the output's going to be in PYU config maker output in the config data store, and then down inside, deep inside that data store. Let's go inside this directory. Once again, you can see there's this data store file, star log. And it has the location of the output for just this config. And if we want to look at it, let's just copy that file name that you do less dash capital S to wrap lines around. And there we go. And MPI exec was just taking some time to start up. But, okay, so I need to change this as config 1, PYU config 1. It's just misspelled. And data store star GFF. There we go. So you can see here we have this file with our final results, and they're mostly EST to genome results. There are also a few protein to genome results and a number of genes. We can quickly count how many genes are in this file. Gene, I'm just going to use a tab character. There are 323 genes in this file, which is more than enough to train with. All right. So my MPI one didn't seem to be slow on. Let's go back to our wiki page. So if the next step is to, let's just take a quick look at this file in JBrowse, see what it looks like. I'm going to zoom out just a little so you can see there's a large number of genes. You might have to click here on the left-hand side to get everything to show up. But here, for example, we have a maker-produced gene. Here we have the EST supporting that gene. And here we have protein evidence. And you can see that maker genes will match either a protein or a EST almost exactly, because that's where these genes are being derived from. So this gene is derived from that protein alignment. This gene is derived from that EST alignment. And many of these will be partial. They will either lack a start or a stop codon because the ESTs or proteins don't align very well. Okay, so this is a good starting point. We have several genes that look like they at least somewhat match the evidence pretty well, and they can be the basis for training a gene predictor. So go back to the wiki page. Now, the next step is we're going to train the program SNAP. SNAP can't read GFF3 gene files. It needs ZFS formats. So what we're going to do is we're going to create ZFF files from these GFF3 files that you can train on. Maker comes with an accessory script to do that. So what I'm going to do is inside this directory, I'm going to create a directory called SNAP. Then I'm going to change into that directory. It's empty. Now I'm going to run GFF3 merge. Before we talked about how that works. I'm just giving it the location of the data store index file, .pyu config1 maker output, so because it takes the data store as a value, and what that did is it merged all contigs into this directory, creating this file form. There was only a single config, so it wasn't an issue. Then the next step is to run maker to ZFF, which is a script for converting maker's output to ZFF format for training SNAP. And you can see maker to ZFF can either take a data store or a GFF3 file. In this case, we're going to give it a GFF3 file. And it runs for just a second. And the output it produces is this genome.annotation file, which is the actual ZFF format. This is what ZFF looks like. You can see for an individual gene, it defines exons, initial, terminal, and interior, and other exons, interior exons, or single exons. And using the models we have in that, we'll be able to train SNAP. Okay. So to train SNAP, these are the commands you run. This is direct out of the SNAP documentation. I'm told Fathom can do a lot more things other than just train SNAP, but I really don't know the other options because it's not highly documented, but it's, at least the training of SNAP is documented well enough. So we can see here we're going to run this first command, which is going to categorize genes as being single exon as having any alternate splicing errors, etc. Then we're going to export these genes onto make them all plus strand. This creates a number of additional files. And then we're going to run this program called Forge, which is actually going to build some data for the HMM to use. And this is running on the plus strand annotations. So you can see there's just a lot of files created here, which is why we created this separate directory. And finally, we're going to run this last command, which is going to create the actual HMM. HMM assembler takes the information from these files and creates an HMM for you. So this PYU is just an ID. It really has no actual effect on the running it. I'm just saying this is the ID for the HMM. Use the current working directory to gather all your data, and then write out to a file that is one directory back called pyu.hm. Once I do that, I can go back one directory, and I have a snap HMM. And that's how easy training snap can be. You can see you run maker to Z of F, then you just run these 4 commands in order, and you produce a snap-based HMM. Okay. Once you have produced that HMM, the next step is to rerun maker. To take advantage of this new HMM file. So here we have our maker options. What we're going to do is we're going to edit these to keep the options the same, except now to use our HMM. So before, let me switch back to the wiki so you can see. Before we had EST to genome and protein to genome on, and we're going to turn both of those off so they don't interfere with our ab initio gene predictor. So I go down to EST to genome, set that equal to 0. Protein to genome, set that equal to 0. And I'm going to put the location of the snap HMM, pyu1.hm. So once we save that, we're going to run maker again. This time we're going to run it in this pyu contig 2, so that way we keep our output separate. And you can see here maker-base, as I copied from here, pyu contig 2. So this is going to write output to this directory, and it also contains some pre-computed data for you. And I do not know what happened with my MPI example. I'll have to figure that out later. Looks like that node died, so we'll just ignore that. Okay, so this will run for probably about 60 to 70 seconds because it's using a lot of pre-computed output, so maker doesn't have to rerun, etc. You can use a closed species for training. Say, for example, you take the entire proteome of a related species. You could use protein to genome to just call genes directly from that proteome. And they're going to be pretty close to your related species. And you'll probably be able to capture enough genes to train quite well on your new genome. In general, you need at least 10 megabases of sequence in many genomes to get enough genes, because you need maybe 400 to 500 genes to train with. But some genomes, like fungi or genomes with higher gene density, you might be able to train in just a couple of contigs that are maybe a couple of megabases in length. And you don't need to train on the entire genome. You can just take a small segment of the genome, call the genes on that, and then use that as the base for your training. There we go. Elapsed time, 83 seconds. And now we've run maker one more time, this time with a training in mind. Rather than looking at the GFF3 again, let's just jump right into JBrowse and see what this other region looks like. Once again, we have, I'm going to click here to make SNAP available. So I have SNAP, my maker transcripts, EST to genome, and protein to genome selected. I have my maker genes here, and you can see the SNAP calls directly below, and the evidence below that. And in many cases, maker and SNAP seem to be pretty close to each other, but in many cases, Snap calls something completely different, or if Snap calls nothing at all, Snap does well here, there's good match, and there's a lot of times where it just doesn't seem to call anything. But Maker still gets, it's still predicting. The reason is because Maker's talking to Snap. It's using, for example, EST evidence down here below to send that evidence back to Snap. And so Snap initially called this, looks like a truncated gene, but the EST suggests it's much longer. Maker talks to Snap, and Snap produces a better model that's much longer and more complete. So as you can see, even we have a roughly trained gene predictor right now, but Maker performs better than Snap alone because it has that feedback with the evidence, where it's able to talk and tell Snap about details that it just can't see otherwise. For example, here, this gene, we have a nice long protein suggesting there's a longer gene on this strand. Snap's calling many small genes. After talking with Snap and showing it the evidence, Snap says, well, maybe it is a bigger gene, and Snap calls it differently. Okay, so because these Maker genes seem to be slightly better than the current Snap genes, we can now retrain using this first set of Maker genes. And supposedly, this should even be better models than our EST genome models, and Snap can use these genes to do even better. So this is called bootstrap training. So back here in the wiki, we're going to basically do just what we did before. We're going to create another directory called Snap2. We're going to go into that directory, copy our GFF3 file output using this command. We're going to convert that output to ZFF format, and then we're going to run Snap's training programs, and that will produce a final file. I'm just going to cut and paste them all at one time. And the result was, of cutting and pasting all of those together, I have my second version of Snap HMM. So this should perform slightly better because it has slightly better models than the first training sets. And in general, 2 rounds of bootstrap training is sufficient to fix this. And all we need to do now is just, instead of using Snap HMM PYU1 in the file, if we edit the control files again, we're just going to change the file. So Emacs Maker Nano is what I use in the wiki. Nano Maker Ops. And Snap HMM, I'm just going to use the new HMM I just produced. That's the only difference. PYU1 is now PYU2. Control O to save, Control X to exit. Okay. Now, if I run Maker one last time, and this time I'd run in PYU config 3, so I keep my output separately. So it's going to write to this third Maker output directory rather than these first 2. So this was the EST genome, protein to genome. The second one was our first run of Snap, and this is our bootstrap retraining of Snap. I'm just going to let that run. The second round usually is much better than the first round, at least with EST to genome. It's markedly different. Sometimes with Sejima, or if you have good training for your first round, the second round isn't worth it. But EST to genome usually tends to be very poor training data. It's just enough to seed Snap and get it to behave. And when Maker talks with it, it gets better models that work for a better training set. But it really has to do more with the fact that we use EST to genome and protein to genome as our base, because they end up being really partial, poor models to begin with. You can train Augustus. There's some scripts Jason States wrote that will actually convert the ZFF training file for Snap and make it into an Augustus training file, which I really like. And then you can train Augustus directly on that output. That way you're using the exact same training data to train Augustus and Snap. You can, I'll try and send an email to the class with the location of his scripts, so you can see how to do that. But there's nothing that comes with Maker that helps. And and this ran in 89 seconds. Now I'll point everyone to where that Augustus training script from Jason Stageroad is. Once the class is over, I'll post it to the class website. All right, so this we ran it a third time. Rather than going to find the GFF3 file, I'm just going to click directly here and take a look at what the results look like this time around. Okay, this time around, we still have things that are different, but in general, there does seem to be a slight bit of less discordance between Maker and Snap. If you remember, I think this exact same gene was being called as 3 genes. Kind of is here, but Snap's picking up longer parts of it. It's this protein evidence that seems to be joining the Snap models. Here we have Snap very closely matching the Maker model and the evidence. Snap very closely matching the Maker evidence. Again. So Snap and Maker in more agreement. So if you were to just run Snap in this genome at this time and not even run Maker, you would probably actually recapture most of the genes almost just as well as Maker does. Maker does a better job because it's able to take the evidence into account, but a well-trained gene finder works well. It's hard to get it trained. Augustus works extremely well when it's trained, as seen by some of its statistics, where you can recapture 80 to 90% of the gene structure just using the HMM across an entire genome. But it's really hard to get to that point. So that's why the Maker model, where you let Maker talk to the program for you, works so well, because it gets around a lot of those, a lot of the hassles of trying to figure out how to get your gene predictor to behave. The Snap seems to find at least a partial model here, and Maker's longer model. It looks like it's partially from here, so I don't know if that might be a false call. There might be some more evidence coming from BLAST-X that I'm not seeing here. Oh, looks like there is. I'm getting a long BLAST-X alignment here. It's suggesting a longer gene for Maker. Okay, but that's generally how you at least get started with the new genome project. You need to produce some rough gene models to train your gene predictor, and then with at least one layer of bootstrap training, you should be able to go with your move towards final annotation. For your initial pass, protein to genome can be better if your organism is closely related to the organism you're aligning proteins with. Many organisms works just fine, especially if your ESTs are partial. Some newer genomes, I know, for example, with lamprey, there was just nothing close to it, and proteins did not align well across species for some reason. So we used mRNA-seq data to derive EST to genome predictions and then use those to train with. And it was really the only way we could get models out of it. And it was just because it was unique enough compared to anything else that had been sequenced, where you just couldn't get protein to genome to work. But I do like protein to genome when you do have a closely related species, because they tend to recover the entire gene and recover it quite well. Okay, so let's go back to the wiki. Okay, looks like we've got a few minutes left. I only have 2 examples to go through. We can take a brief pause for a couple of minutes, making sure all of the questions are being answered. I know it's sometimes hard to follow along, but if, like I said, if you're having problems with the filling out the values, you can do something like the following: copy ops to Maker ops. So if your first attempt to edit the file fails, just copy it across and then run Maker that way. Base, and you can see, I just run that for a second, you can see, oh, it's already done. But if you also change Maker options, because Maker reuses its output, if we actually use the same directory instead of 3 separate directories, what Maker would do is it would look inside this directory, delete any output it thought was no longer relevant, and it would regenerate it would reuse any output that it thought it could reuse because it wasn't changed by the way you manipulated the option. So rerunning Maker can actually be much faster than running Maker directly. And that's part of the theory with why I have you unload this partial file, because Maker's smart enough to know that those are the output files it's going to need, so when it sees them, it just reads them rather than rerunning BLAST. Okay, so let's go back one directory. So this next example is for legacy annotation pass-through. This might be where you have an existing genome. You might have multiple data sets. For example, you might have 3 legacy annotation data sets, and you want to be able to merge them into a non-redundant new data set. And you might want to be able to update the data set in light of new evidence, mRNA-seq data, etc. So give it a chance to accept the models as they are, or replace them at any given locus if the evidence suggests it. And Maker can do this. It uses, to get these legacy annotations into Maker, Maker uses what's called GFF3 pass-through. Here are several options you can see here, where GFF3 pass-through is used for getting different types of evidence into Maker. For example, you can get EFT evidence from things like couplings or top hat into Maker using this option. You can, if you have protein evidence, you might have aligned it with BLAT, and you convert it to GFF3. You can give it here. For ab initio gene predictors, if you have a gene ID or GenScan or any gene predictor that Maker doesn't support, if its output, if you can convert its output to GFF3 format, you can give it to this option, and Maker will allow it to compete. Then model GFF, this is where you can give Maker pre-existing annotation, things that you want to be kept even if there isn't any evidence to support them, but allow Maker to replace those annotations if there's evidence suggesting that there needs to be a different model. And that's what we're going to use today, is the model GFF option. So once again, let's get started. You can see here we change to this directory and look inside. We have, once again, this is the output that will be there when we finish running Maker. This is partial output where we can pre-populate the directory so we don't have to run BLAST, repeat master, etc. I'm actually going to have you do that. tar-dxf partial. You're right. And that will create this output directory that's at least pre-populated with some of our output, so it will run really fast. Because this is also a big config, about 2 megabases in length. To run this, we have several files here. We have the Pythia multum ESTs that we used in our last analysis, SP protein. We also have the genome config, but we have 2 GFF3 files with legacy annotations. Take a look at those. Oops, legacy set 2. Let's do, I'm going to use a dash capital S so the lines wrap around. You can see here we have, these are just hypothetical legacy annotations called legacy set 2 gene so-and-so. And here would be legacy set 1, genes as well. And we also want to be able to run SNAP, so I have a Pythia multum ATMM training file, just like we just made, that we're going to let SNAP replace any of these models if SNAP finds something better. So first we run maker-ctl to create the control files. Now this is the control file. We always edit this maker options file. You see here, or you can just copy this options file to maker ops if you don't want to edit it. We're going to change the following options: genome, EST, protein, maker GFF, and SNAP HMA. So let's get started. Nano maker ops.ctl. So the genome needs to be legacy config.fasta. EST needs to be PYU EST.FASTA, and protein needs to be, just as in the previous example, SP protein.FASTA. Okay, now here's, let's see, let's put it a little further. We hit snap HMM. That needs to be PYU.HMM. And we need to find model GFF, and that's down here below. So these are the two legacy annotation sets we're going to be handing in. And notice I'm using a comma-separated list here. So the comma-separated list is a maker feature. You can, any of these options can take it, actually. If you have multiple data sets and you don't want to provide a single file, you can just put a comma on there and add a second file. So you can see we're going to have two legacy annotation sets. We could have 6 protein data sets. You could have EST data sets with multiple tissues and treatments if you want. You could even have multiple HMMs if you wanted and run snap 2 times with multiple HMMs. I've never done that, but you could. Another option that Maker has that I might as well talk about while we were talking about the comma-separated list, Maker also has a label option I talk about here below. If you can see here, if I use a colon, I can actually add a label to these files. That's nice because if you were using a comma-separated list, you might want to be able to distinguish output from one file to the other. For example, here, hypoxia, these might be ESTs collected from a low oxygen study. Well, the result would be in the GFF3 file, when you look at the column that says what source the data came from, it would say something like EST to genome, but if you have the label, it would say EST to genome hypoxia. Many GMOD programs can use this, like gbrowse, to provide additional on-off switches that let you turn off just some of your ESTs or just some of your proteins. You could, for example, it would turn off just, you get a switch that would turn off just hypoxia ESTs. And it also gives you just a nice different labeling tag. So if you were using jbrowse, you can give them a separate tier to other ESTs. And, you know, just for kicks, I'm going to go ahead and add this onto the results. Okay. EST. Up here, I'm going to go ahead and add hypoxia on it just for fun. This is optional. You don't have to do it if you don't want to, but it'll let you show what the output looks like when you have a label. Okay. So Control-O to save, Control-X to exit. Now I have my maker options. Now we are ready to run. Check something out. Okay, this is my other instance. What I'm doing here, I just want to check CTL. I want to once again try the MPI option just to see if it will function this time. Okay. CP ops to maker ops. Okay. And if we run, okay, I'm going to run an MPI exec. I'm going to run maker here on this one. And here is the other one where we're going to run. I've already unloaded the partial data. I just have to type maker, and this will run. And so yes, you can for the GFF3 files, the labels are actually in the GFF3 file. So the labels really only affect the FASTA file options, but say for example we were to, if you look in one of these files, these already have labels. They are legacy 1. So by default, Maker will use these as the labels. So you'll get results in your Maker annotation file that will contain model GFF colon legacy 1. Well, what I should do is I should let you add a colon and have your label override this label. I just haven't done that. Okay, so I'll type Maker. Take a moment to run. I just want to check this other one. No, MPI just doesn't want to work for me for some reason. So this will take probably about 80 seconds to run. Like I said, we pre-computed our output. Okay, 52 seconds, not too bad. I'm going to go ahead and look inside this file, go all the way down into the legacy data store, and if we look down inside this deep directory, we see our GFF3 file there. I'm just going to open that up so we can take a look and see if we actually do get our hypoxia label. Les-S, that big long file name. There it is. As you can see, EST to genome now has hypoxia on its label. And if we go, we can look at the output in JBrowse. This doesn't, I don't know if I ran the hypoxia label in this example. No, I didn't. Source is EST genome. So when I ran it, at least for JBrowse, loaded in GBrowse, I didn't have the hypoxia label turned on. But it's the same thing. You can see these are the models we just produced. I'm turning on SNP, transcripts, EST to genome, and protein to genome. I got here by clicking on C example 3. So this is the results of the GFF3 file we just produced. And you can see here, these are our final models, and these were our input models. Oh, I'm going to turn on this too as well. So these are our 2 legacy annotation models. Let's move these up higher so you can see those. Okay, so I have my 2 legacy models, my SNP models, my maker transcripts, EST genome, and protein genome evidence. So here we can see our legacy model is similar to SNP, a legacy model 1. Legacy model 2 is very different though. And in this case, it decided to go with legacy model 2, probably because it better matches this protein evidence. Here's an example where the legacy model 1 was kept. This is probably because it's so similar to the SNP model. It does its best to keep legacy models whenever possible, because it considers the fact that there are already annotations as at least a certain level of support that it should exist. Here's a case where it actually new model was either created or it overrode 2 legacy models. Maker thought the SNP model slightly better or represented the evidence alignment. There's probably a BLAST X alignment down below. There we are. That aligns well with SNP. I'm imagining it has a slightly better overlap with the BLAST X evidence than either of the legacy models, which is why it was kept. And so on and so forth. So you can keep going through here, and you can see there's we now have a single non-redundant transcript set that was created by merging the legacy models, legacies 1 and 2, and allowing SNP to replace or edit things wherever possible, as shown. So this is one way to reannotate genomes. You can also, if I provided these as model GFF, if you provide them to the PRED GFF, you get virtually the same result. The difference for PRED GFF is that you don't keep the model if there's no support, but model GFF will still keep the model if there's no evidence to support. So it's a way of making sure you don't drop an annotation from your legacy data set just because Maker couldn't find an evidence alignment. Okay. So here's just a few examples of how this is working and why it can be used. So the AED score, as I explained before, ranges from 0 to 1, 0 being a perfect match, 1 to a poor match. This is a figure from a previous paper where we're looking at the mouse genome where we just passed genes through and didn't let Maker change them and just scored them with AED. And you can see we have mouse release 30 and mouse release 37. And we have a cumulative distribution of the AED scores for those releases. And anything further pushed out towards the left corner is better annotated than anything down here to the right. And you can see Maker shows that 37.1 is better annotated than release 30, which was expected. The dotted lines represent ref-seq genomes. So ref-seq genes tend to be better evidence supported than the genome as a whole. Annotations with low AED scores tend to be enriched with protein domains. High AED scores tend to be poorly enriched for protein domains. And this section here, where we're looking at presence or absence, we show that anything with a low AED score tends to have been kept in both releases, where anything with high AED scores was actually thrown out by the manual annotators between release 30 and release 37. This is just showing how AED score can overall mimic some of the statistics used by manual annotators for deciding annotation quality. And it's these same AED scores that end up being used to help guide the re-annotation of whether a gene model should be kept, dropped, or updated with new evidence. Here's an example of re-annotation in maize, where we have the maize genome. We have a de novo genome using Maker, and then a pass-through where we let old maize annotations come through, so we keep them even if they have no evidence support, which is why you get this bump here. And we have a gold standard of well-annotated, well-supported maize genes. And in general, you can see that when passed through Maker, you get better annotations, much more like the gold standard. And if you do a de novo annotation, it's very similar to the gold standard. But with the pass-through option, you keep these unsupported genes because just because there's no evidence, you don't necessarily want to have Maker throw them out. Okay, so once again, the difference between model GFF and pred-GFF. They both are gene predictions. Pred-GFF can make it into your final gene annotations if it's the best scoring model and only if it has evidence support. So if it has no support, it won't be kept. A model GFF will always be kept if it's either the best model or nothing used to support it, but I'm not going to get rid of it because it was in your old annotation set. So you keep it anyway. This very last example will take just about 10 minutes. It goes really fast. No, it's a very long day. So this is when you've finished genome annotation, you finally have your final data set. The next question is, what did the genes do? I'd like to have some extra information in the gene annotations that make them more useful to me as a researcher. So this last example is using some Maker accessory scripts to add some extra data. So we're going to go ahead and change to Maker tutorial example 4 post annotation. It's seen here. And let's go ahead and look inside that directory. And you can see there's a number of files. Once again, you have a finished file for what we should have when we're done. And what we have is an already existing GFF3 file that would represent your Maker-produced final merged GFF3, a protein file and a transcript file representing all the annotations of the genome. But in addition, we've run a few analyses. I won't explain how we ran them. I'll leave that as an exercise for you. We ran a BLAST report. This is a tab-delimited, capital S, tab-delimited BLAST report where we blasted the Maker genes against UniProt, SwissProt. And you can see we can see the best hits for each Maker gene, and this tab-delimited report are here with their e-values and some other statistics calculated by BLAST. And here we have, for its -M format 8 in NCBI, if you want to know how to create a tab-delimited BLAST report, it's just one of the command line flags in BLAST. And then this is an inter-proscan report. So we have the Maker genes, and then the inter-proscan identifies protein domains within those genes. And you can see Panther, PFAM, and if you go out here, it also identifies extra information such as Go terms, potential functions for those genes. So I'd like to be able to include all of this other data that I got from these additional programs that run directly on the protein FASTA file and integrate it into the annotations. So, and another thing I'd like to do, I'd like to change the really ugly Maker-produced names. Maker produces names that are informative, at least at first, to tell you where the gene came from. This came from SNAP. It's located on this contig. It was the first gene on the contig to be produced, and it's mRNA1, but it's a really long, ugly name that you don't necessarily want to keep when you get ready to submit your genes. So we have a program called Maker Map IDs that allows you to rename genes in a more NCBI suggested format. Let's just run that so you can see the usage statement. And you can see it takes a prefix, which would be a prefix you might have registered with NCBI, and you can then create a digit prefix, a digital ID that's 8 characters in length. So we're going to create a prefix called GMOD and 8 characters wide, and save it in this mapping file. Okay, I just ran that command, and I created this file called hstat.config.map. Let's take a look at it. And you can see it's just a 2-column file. This is the old name, and this is a much friendlier new name. Old name for the mRNA, much friendlier new name for the mRNA. And there's lots of options where you can change the suffix and the prefix and use different formats for naming genes and mRNAs that are included in that command. So there are 3 scripts in Maker that can read the map ID to do conversions of gene names. There's map FASTA IDs. This fixes names in FASTA files. Map GFF3 IDs. This fixes names in GFF3 files, and it also adds an alias attribute so you can recover the old names if you want. And map data IDs. That tries to just replace names in any generic data file that you have. It does its best. It doesn't always work, but it does a pretty good job. So because these scripts do in-place replacement, it's best to copy the file to a new location before you run any of these scripts. So that's what these copy commands here, where I'm just copying config.gff to config rename.gff. Same with the FASTA files. I'm adding a renamed suffix and output.iPrScan to output rename.iPrScan. Okay, so I run all those commands, and now I have a whole bunch of new files that are renamed. And just so you can see that they were really named, I'll go ahead and open one. You can, run. Maybe that one of my commands failed. Just give me a second. Okay, so that is, all I did was the copy, sorry. So all we did was copying right now. The next step is to run the replacements. We're going to run map GFF to replace the GFF IDs, map FASTA to replace the FASTA IDs, and map data IDs on these 2 output reports to replace those IDs. I run all 4 commands, and now this renamed GFF3 file should use the new names. Let's use a -S so lines wrap around. And you see we got much nicer names. ID, GMOD1, name GMOD1, and then we have the old maker name and the alias. And we also get the same with our output reports. Oops, -S. We see that the old maker, a new maker name is now there in the output reports of the old maker name. So we can use those output reports. All right. Now the next step is to try and add the content of these output reports to our Maker annotations. We have a Maker functional GFF. This takes blast reports where you've blasted your Maker genes against a UniProt SwissProt database, and it will add the putative functions to Maker GFF3 files. And Maker functional FASTA does the same, except it adds putative functions to the FASTA files using the UniProt SwissProt database, the blast report, and the FASTA files input. So I'll go ahead and run these commands to change the information contained within the Maker GFF3 file and both Maker FASTA files. And the result should be a new file, let's do ls-1 so they fit on the screen. As you can see, we have the putative function added to the FASTA files. Let's just open one up so you can see what it looks like. Okay, Maker GMOD RA1 protein name similar to Run DC1 from Homo sapiens. So this is a nice way to use a well annotated database like SwissProt to try and at least infer some similarity to other gene types. Okay, then the next one we're going to look at is InterProScan output, the main output. Yes, the options do not override the GFF3 output because there's already a label in column 2, and Maker uses that label. I probably should edit it so that if the user supplies a label, they use the user's label as opposed to the label in the GFF3 file. It's column 2 in the GFF3 file. And you'll notice that in column 2 of legacy set 1, there's actually something called legacy 1. And in column 2 of legacy set 2, there's a legacy 2 label. And those labels will be applied to the model GFF attribute in those files. I'll show you that in just a moment. I'll show that in just a moment. Okay, so the next step in this was to add the InterProScan output. So there's 2 scripts. There's IPR update GFF. This will just take the InterProScan output file and just add the tags of what domains are contained within the gene to the gene, so that you can search for genes containing certain domains, for example. Then the second one, IPRScan to GFF3, this one creates physically viewable features. You can actually view them in JBrowse to see what the domain looks like within the gene. So I'm just going to run both these commands real quick. All right, and it produced, let's look at one. Okay. We can see this where we've added the domain to the GFF3 file. I'll let you see what that looks like. Now, you can see here, we have the domains added as dbxref, interpro domain IDs, Panther domain IDs, smart, superfamily, pfam, etc. In addition, we've also been able to infer from those domains Go terms. Let's find an example where we have a Go term. Here we are, ontology term, and these Go terms are now also associated with that gene as putative either function, locations, or molecular process, etc. of the that particular gene. And in this visible IPRScan domains.gff we just created, you can see that we have IPRScan features, match parts, that will be viewable in the browser that actually map to the structure of the domain against the gene. And to explain how that looks, we can click on the before and after of what this looked like before. So this is what this region of the genome looked like before. These are the gene models. These are protein evidence alignments supporting the gene models. And you can see that we have these long, ugly maker names. And if we click on one of these, there's really not a lot of information extra contained within the model. There's no putative function. There is an AED score telling us how well it matches the evidence, but not a lot else. But after we've run the functional annotation accessory scripts, we now have much nicer gene names. We have putative gene functions showing up in the browser. If I click on one of these, you can see these are the domains contained within the gene, dbxref. These are the domains contained within the gene. This particular one doesn't seem to have any ontology terms. Let's pick on a different one. Like this one. And yep, these are Go terms associated with the gene. So you get much richer data within your genomic database. And if I click IPRScan, I'm going to bring this up here so you can view it. These are the InterProScan domains actually mapped against the gene so you can see where the domains occur. And you can see this is a run domain in the run-DC domain containing protein, similar to ribosome protein L26, and this is ribosome protein L27E protein domain, you can see which sections of the gene now correspond to which domains and subdomains. And this may give you much, much richer information than if you were designing an experiment, for example, targeting, this kind of information in your database would give you a lot better available to put this into experimentation. For example, if I wanted to try and knock out or affect individual domains, I now know where that domain is located. And I could identify all the in that domain. Okay. So I'm going to go back to one pending question about column 2 from the last example. Let's take a look at the GFF3 file so you can see that. Legacy, data store, and the GFF3 file. Oops. Here we are. So you can see that the reference annotations from the legacy pass-through is model GFF legacy 1. So they're still in the file as a reference with the recording labels from column 2, but the labels aren't carried over to the file annotations. They're just there in the reference match part. So it's about 5:20 in the East Coast now, so you've probably all been saturated with information on annotations, GFF3 files, etc. The only last thing I'll leave you with here, brief discussion of maker accessory scripts. We've already used a lot of these. There are additional scripts that, like Chatto2 GFF3, that help you create GFF3 files if you have a Chatto database. Or I also have a GFF3 Chatto that can do the exact opposite, dump your GFF3 into Chatto. Cufflinks to GFF3 is a nice one for cufflinks. FASTA tool is a nice tool just for manipulating FASTA files, splitting them, searching for individual IDs. And this maker to JBrowse is now part of JBrowse and helps load maker's output into JBrowse, maker to Chatto, load it into Chatto database, and that's, and Top Hat to GFF is another very useful one of these accessory scripts.